Record na ba? And okay. So again, today our topic is on DNA replication. So let's start. Mhm. First, let's look at the central dogma of molecular biology again. So, di ba last time we discussed about the DNA structure and the hereditary information. So, dito tayo na part ng central dogma of molecular biology. And now, we will discuss DNA replication or the formation of DNA from another DNA. So, we will discuss this in depth and in great detail. So, sundan nyo lang ako sa lecture na to. So, ayan. DNA replication. So, bakit nga ba nagkakaroon ng DNA replication? So, hindi dahil sa Kagebunshin no Jutsu ni Naruto. Parang gano'n lang naman yung principle para magre-replicate yung DNA mo. Parang multiple copies of your DNA. But the purpose of DNA replication is to pass on your hereditary information from one cell to another. So, if you remember in our lecture 1, right, yung nagdi-divide na cells, 'di ba? Para magkaroon ng division of cells, yung hereditary information mo, you have to pass it on to your daughter cells. So, yun yung purpose ng DNA replication. Okay? So, the objectives for this lecture is to discuss the changes that occur in the DNA as it is passed down from generation to generation. Next, to learn the mechanism behind DNA replication. And lastly, to understand the principle behind a laboratory technique using the mechanism of DNA replication. Dinidighay ako kasi kakakain ko lang ng breakfast. Sorry guys. So, yun. Yun. So, first, let's look at the maintenance of DNA sequences. So, naalala nyo to sa lecture 1, right? Ito yung tree of life. And we have discussed in our previous lecture that genetic changes throughout the tree of life gives every living organism some kind of variation, right? And these variations, they enhance the survival of each species. So, kaya nagkakaroon, dahil sa genetic variation, each of these species, they have more chance to survive. Okay? Yun yung advantage ng genetic variation in the level of the species. However, in the level of one individual organism, we want to avoid mutation. Why? Because if you, an individual organism, you have a mutation in your DNA, it may cause some diseases such as cancer. So we want to avoid that. Okay. So now, since we want to avoid these mutations, we will take a look at how our cells during DNA replication avoid having these mutations. Okay, so throughout this lecture, you will have an idea on how our cells they avoid for us to have mutations in our DNA. Okay. So, but first, let's take a look at mutation rate. So again, this is the rate at which changes occur in the DNA sequences. So we have established, right, that every organism, they mutate. And these genetic variations, they pose a great chance for the species to survive. However, in the level of an organism, we want to avoid a mutation kasi pag nagkaroon ka ng mutation sa DNA mo, pwede ka magkasakit, right? So, we now study the mutation rates, and these mutation rates, it is just the rate at which changes occur in our DNA sequences. Okay? Sabihan niyo kapag mabilis ako kasi minsan mabilis pala ako magsalita tapos di nyo na naiintindihan. Okay. So, ngayon, E. coli, yung model organism natin for studying mutation rates. And why? Because E. coli is a bacteria that can give rise to a large population in less than a day. Actually, kahit anong bacteria, mag-give rise ng a lot of population in less than a day. But yung model organism natin ay E. coli. Excuse me. So, E. coli can give rise to billions of cells in less than a day. So, if you remember in your statistics class, nag-start na ba kayo? Sa statistics class nyo, right? Kailangan nyo ng large population size for your, to observe if your variable is significant, right? So, that's why we can see the mutation rates in E. coli because, again, E. coli can give rise to a large population. And in that population, you can see a fraction in which they mutated. So, yung fraction ng population sa E. coli na yon, makakakita ka ng fraction sa population na yon na nagkaroon ng mutation. So, how? Of course, you do an experiment. So, everything in science, to prove to answer a question in science, you always do an experiment, right? So, ito yung naging experiment on mutation rate. So, for example, you are you want to observe the mutation rate for the gene of E. coli to metabolize lactose. Okay? You want to observe the mutation rate for the gene to metabolize lactose. So, inherently, E. coli has the gene to metabolize lactose. Okay? Okay. So, now, what you will do is to grow your E. coli in a plate without lactose. Okay? So, yung plate na yon ay glucose only. So, you you let your E. coli here grow in your glucose only plate, and after some time, some fraction of the population of the E. coli will mutate to have a defective gene to metabolize lactose. So, how will you see that? Yung di yung yung pinag-grow mo dito sa plate na to, you let it grow again sa plate na may lactose only. So, you will observe that some of the colonies here did not grow. Why? Again, because nagkaroon ng mutation sa gene to metabolize lactose. So, nung nilagay mo na siya doon sa plate na meron lang na lactose, nawala na siya ng ability to metabolize lactose. Okay? So, yun yung mutation. Nagkaroon siya ng mutation na hindi kaaya-aya. So, some portion of the population, nagkaroon siya ng mutation. And now, you can calculate your mutation rate. But, you have to also take into account silent mutations. So, itong mutation dito na nakita natin, observable mutation, right? Because na-observe mo na yung ibang colonies, hindi na sila nag-grow. Therefore, nagkaroon sila ng observable na defective yung gene nila for lactose metabolism, right? But, you also have to take into account some silent mutations. So, what are silent mutations? Silent mutations are changes in your DNA sequences na hindi naman nagkakaroon ng effect sa organism, sa characteristic ng organism. So, silent lang siya. Parang, for example, nag-iba yung base mo ng A, from A naging T, ganon. Pero hindi naman na-affect yung kakayahan niya to metabolize lactose. So, those are silent mutations. But we will discuss silent mutations further in our next lectures para mas maintindihan nyo at mas ma-appreciate nyo ang silent mutations. But ngayon, now that you have to take into account your silent mutations and your observable mutation, you can now compute for your mutation rate. And di nyo kailangan to tandaan na numbers, but like the mutation rate for E. coli is about 3 mutations for a billion of nucleotides per cell generation. So, in relatively, this mutation rate is actually low. Low mutation rate lang siya kasi 3 mutations lang per a billion of nucleotide. So, per a billion of nucleotide, isipin nyo, isang billion ng nucleotide. Tapos 3 lang dun yung magbabago. Diba? Sobrang onti lang. So, low mutation rate lang siya actually. Okay? And in humans, in comparison, the mutation rate is about one mutation per billion of nucleotides per cell division. And as you can see, mas mababa, right? Or halos same lang. But again, this is considered as a low mutation. So, ma-appreciate nyo tong mutation rates mamaya pag diniscuss natin yung DNA replication. So, tandaan nyo lang na yung E. coli and yung humans, they have relatively low mutation rates. Okay. And now, we go to DNA templating. So, alalahanin nyo sa lecture 1, si Watson and Crick, right? Rewind, lecture 1. Watson and Crick, inaral nila yung DNA structure. And actually, Watson and Crick, they also gave us the idea on how DNA replicates. Actually, sa paper nila on DNA structure, sinulat na nila doon kung paano nagre-replicate daw yung DNA. So, sabi nila, if you remember this figure, each strand daw of the DNA, you can use them as template. So, if you use this as strand, the 5' to 3' strand, you can create a new S' strand. And similarly, if you use this S' strand, you can just create a new S strand. So, yun lang naman yung ibig sabihin ng DNA templating. Kahit ano daw na strand dito sa DNA mo, pwede mong magamit to create new DNA. Okay? So, kaya important na inaral ni na Watson and Crick yung structure ng DNA kasi actually, yun yung parang stepping stone para maintindihan natin yung how DNA replicates. Kasi kung di nila yun inaral, iniisip pa rin natin ngayon kung paano nagre-replicate ang DNA actually. Kaya important yung findings nila. Okay? And now, you will observe that DNA is actually semi-conservative or DNA replication is actually semi-conservative. So how? The daughter cells, they inherit one original strand and one new strand. So observe nyo ulit. Itong figure na to, right? Yung daughter cells daw, mag-inherit daw ng one original strand and one new strand. So let's take a look. Ito yung parent DNA mo, right? Ito yung parent DNA mo from your parent cell. So now, when this parent cell divides into two daughter cells, yung daughter cells daw mag-inherit ng one original strand, right? So, di ba? Ito yan. Itong nasa taas. And one new strand. So, ito yung new strand. So, similarly, yung isang daughter cell mo, yung isang daughter DNA mo, mag-inherit din ng one original strand and one new strand. Right? So, kaya daw, semi-conservative ang DNA replication. So, ito pa, para ma-establish natin. So, as you can see, right, as the generation goes further, yung one daughter cell or one daughter DNA, nag-inherit siya ng original strand and new strand. So, take a look at this first generation first. So, ito yung parent, di ba? Yung daughter cell niya, itong isa at ito pang isa. Yung isa, naka-inherit ng one original strand, yung yellow, and new DNA strand, yung red. And similarly sa isa pa. So, if you take a look again at this generation, yung daughter nito, makaka-inherit ng original strand, red and yellow, di ba? Yung original dito, red and yellow, and new DNA strand, yung green. So, yun lang naman yung ibig sabihin ng semi-conservative, okay? Okay. So, let's move on. So, DNA replication. There are 3 steps in DNA replication. So, first is initiation, second is elongation, and third is termination. So, yan yung basic steps in DNA replication. So, first, let's take a look at the actors. Wow, table, di ba? Wow. Ma'am, seryoso ka? Ba't ka naglagay ng table dyan? So, bakit ba ako naglagay ng table dyan? Gusto ko lang i-list yung actors natin. So, pag nag-aaral ka ng DNA replication, actually, isipin mo, nanonood ka lang ng K-drama. Okay? Paano ba? Kasi sa DNA replication, same lang yung actors mo. Same actors, pero different names. Pero same lang rin sila ng roles. Okay, para ma-appreciate nyo. Yan, naglagay ako ng Boys Over Flowers at Meet Your Garden, di ba? So, di ba sa Boys Over Flowers, si Goom Jandi, same lang naman siya ng role kay Sunshine. Pero, different yung names nila. So, ganun lang naman yung DNA replication din. Parang, yung enzymes and proteins mo, same lang naman yung roles nila, pero nag-iiba lang minsan yung name nila for eukaryotes and bacteria. So, for example, sinabi ko 'di ba si Gumjandi sa Boys Over Flowers pero si Sunshine sa Meet Your Garden. Same role, different names. 'Yon. Okay. So, for example dito, tingnan natin. Example lang, aaralin naman natin ito. For example, in bacteria, you have DNA gyrase. And the counterpart of DNA gyrase in eukaryotes is topoisomerase. And what it does is that it just unwinds your DNA. So, parang inaalis niya yung pilipit. Malalaman natin bakit mamaya. So, yun. And another example ay yung DNA polymerase mo. So, in bacteria, you have DNA polymerase 1 and 3. But in eukaryotes, you have alpha, gamma, delta, and epsilon. So, actually, halos same lang yung function nila, pero different lang yung names for bacteria and eukaryote. So, kapag mag-aaral kayo again ng DNA replication, isipin nyo, same lang yung characters, different lang yung names nila, pero same lang yung roles nila, ganun. Okay? Yan yung trick. So, ngayon, let us take a look at the main characters for our drama. So, okay, yung drama natin, DNA replication, right? Ito yung main character. So, for simplicity, yung sa bacteria yung titingnan natin. Kasi, halos same lang naman yung function nila sa eukaryotes. Okay, so ito, yung first natin na character is DNA gyrase. So, DNA gyrase, here yung pink, it is responsible for unwinding the DNA. So, 'di ba, DNA is a twisted ladder. So, for you to stretch your DNA, kailangan mo siyang i-untwist. Tapos, yung responsible doon ay yung DNA gyrase. And yung counterpart nito sa eukaryotes ay yung topoisomerase. Okay? Overview lang to. So, yung next main character natin is yung helicase. And, meron bang question? Wait. Yung next character natin ay yung helicase, itong purple. So, yung helicase mo, yung main role niya sa drama natin ay magse-separate siya ng DNA. Okay? It basically unzips your double-stranded DNA. Sineseparate niya yung dalawang strands. Okay? And yung next character, itong gray na to na bilog-bilog, ay yung SSB proteins or single-stranded binding proteins. Yung role lang nila ay to hold each of the strands together to prevent them from re-annealing. So what is re-annealing? Re-annealing basically magdidikit ulit. So para 'di ba, hindi magdikit itong isang strand dito sa isang strand ulit, kailangan sila i-hold ng SSB proteins, itong gray. Okay? And yung next character natin ay ito, DNA primase, and it makes the primer. So, your primer is composed of RNA, and your primer marks the start of DNA replication or where DNA replication starts. Okay. And yung next character mo ay yung DNA polymerase. So, in bacteria, it is DNA polymerase 3. And DNA polymerase 3 is responsible for extending your DNA strand. So as you can see here, itong strand na to, 'di ba? Continuously na ine-extend siya ng DNA polymerase. And this is your leading strand. And the other strand is called your lagging strand. So it is replicated in segments. Okay. And your next character is your DNA polymerase 1. So your DNA polymerase 1 mo, it removes your DNA primer. Because again, DNA primer is made up of RNA. And yung ginagawa natin ay DNA. Okay? And lastly, yung last character mo sa drama natin ay yung DNA ligase. And DNA ligase just basically joins the fragments together in the lagging strand. So, in-depth natin i-discuss each character sa lecture na to. Overview lang yun. So, para lang makilala nyo na sila. Okay? Wait lang. Wait lang. Anong nangyayari? Wait. So, let's take a look at initiation. So, how does DNA replication start? It starts in the replication origin. So, ano ba yung nasa replication origin? It is the binding site for a large multi-subunit initiator protein called the ORC, or origin of replication complex, and a stretch of DNA that is rich in adenines and thymines. So, let's take a look. So, sa origin of replication mo daw, sa replication origin mo, ito, meron daw magbabind na initiator protein called ORC. So, ito yung ORC mo. And this is your origin of replication. Okay? So, yung ginagawa niya, dun nagmamark kung saan magsa-start yung DNA replication mo. And now, it is a stretch of DNA that is rich in adenines and thymines. And bakit kaya rich in adenines and thymines? So, di ba? Bakit kaya rich in adenines and thymines? Meron bang nakakaalam? Or meron ba kayong naiisip kung bakit adenines and thymines? Bakit hindi guanine at cytosine? Bakit kaya? Hmm. Bakit kaya yung origin of replication rich in adenines and thymines at hindi cytosine at guanine? So, yeah. um, Rich in adenines and thymines dahil, remember in your lecture 1, remember nyo yung structure of the DNA, right? So, adenines and thymines are made up of 2
Two.
hydrogen bonds. 2. And guanines and thymines are made up of 3 hydrogen bonds. Okay? So, again, adenines and thymines are made up of 2 hydrogen bonds. And guanines and cytosine are made up of 3 hydrogen bonds pag nag-bind sila together. So, kaya, adenines and thymines ang nasa origin of replication kasi mas madali silang ma-break. Dahil, dalawa lang yung hydrogen bonds nila as compared to guanines and cytosine, right? So, yun yung purpose kung bakit natin inaral yung structure ng DNA para malaman nyo kung ano yung mga details doon at yung mechanism behind everything. So, adenines and thymines again has 2 hydrogen bonds between them. Therefore, they are relatively easier to break than guanines and cytosines. Okay? So, yun. And now that let's take a look at this figure. Ito yung stretch of DNA mo, right? So, sa replication origin daw, sa area na to, rich in adenines and thymines. And then, your initiator protein called ORC, or origin of replication complex, itong orange, magbabind dito. Thus, opens the double helix. And then, your DNA replication proceeds. So, ito lang yung overview niya. But then, as you can see, there are two forks, right? Merong dalawang forks na na-form. And ito yung fork 1 and ito yung fork 2. So, tingnan natin. Again, the replication fork, ito yung sa bacteria because bacteria has a circular DNA, remember? So, now, ganito yung itsura ng replication fork sa bacteria. So, there are two replication forks. Dito, nag-open yung DNA at nagsa-start yung DNA replication. So, the two replication forks, they move in opposite directions. Okay? One goes here and the other goes here. Yun, di ba? Hanggang sa ma-replicate yung buong circular DNA. And ito lang, picture lang nung DNA ng E. coli. So again, there are two replication forks. Okay? Ayun. So, when we take a closer look at the replication fork, ito yung itsura niya. So, kung titingin ka sa isang replication fork, ito yung itsura niya. And ito na makikita mo yung leading strand and yung lagging strand. And last time, di ba, nagtanong si Leila, ano daw yung leading strand and ano yung lagging strand. So, sa replication fork, makikita nyo na yung leading strand, continuous yung pag-replicate ng DNA or paggawa ng new DNA, and it is towards the fork. Meanwhile, the lagging strand, discontinuous or the DNA synthesized in segments. And these segments are called the Okazaki fragments. Okay? So, why are they named Okazaki fragments? Konting history lang. Yung naka-discover nito are mag-asawa na ang apelyido nila Okazaki. And they are a Japanese couple. So, yun. So, yung leading strand mo, ito, di ba? And ito yung lagging strand. So, bakit kaya yung isa continuous at yung isa in segment? Bakit kaya? So, resume or recall nyo sa lecture 1 nyo, nucleotides, they only add from the 5' to 3' direction, right? So, sa 3' ka lang makaka-add ng new nucleotide base. And your DNA strand is anti-parallel to each other. So, since anti-parallel ito, di ba, 3' to 5', 5' to 3', anti-parallel sila, di ba? Now, therefore, yung magagawa mong new strand, yung isa, continuous from 5' to 3' kasi yung template niya, 3' to 5', and yung ginagawa niya ay complementary doon sa parent template DNA. Okay? Now, yung isa mo na anti-parallel, 5' to 3'. And yung gagawin mong DNA, new DNA ay complementary, right? So, 3' to 5' ito dapat. But then again, yung addition lang ng nucleotide bases mo, 5' to 3'. So, therefore, this strand, called your lagging strand, is synthesized in segments. Okay, and we will take a look at the chemistry behind the 5' to 3' mamaya sa elongation. So, initiation pa lang to. Okay. Now, yung pinakita ko dito, yung sa prokaryotes lang, di ba? So, circular DNA lang. But how about in eukaryotes? So, ganito yung itsura sa eukaryotes. There are multiple replicons. So, there are multiple replication origins. So, again, remember in your lecture 1 that the DNA, when you stretch it, right, 2 meters, so linear yung DNA natin. But in bacteria, circular. Yung sa atin, linear. So, how do you replicate a linear DNA? So, ganito yung itsura. Merong multiple replication origins. And these replication origins, as they progress, they combine with each other until all of these fuses together and thus creating now your new DNA strands. Okay? So, ganyan yung itsura kapag linear yung DNA mo sa eukaryotes. in comparison to the circular DNA in bacteria. Okay, so now let's take a look at the proteins involved in unwinding the DNA at the replication fork. So, ito lang yung main characters natin muna. So, yung first ay yung topoisomerase. And topoisomerase, ang counterpart niya sa bacteria ay gyrase, okay? And it unwinds your DNA helix. And the next is your DNA helicase, which unzips your DNA and your single-stranded DNA binding proteins or SSB proteins na humahawak doon sa separate DNA strands mo. Okay? So, DNA topoisomerase muna. So, bakit kailangan natin ng DNA topoisomerase? Nakikita niyo ba yung figure? Gumagalaw ba? Pakigalaw ang baso. Pwede po pa thumbs up. Um, okay. So, kapag, remember in your lecture 1, yung DNA, di ba, twisted ladder. Twisted ladder siya. So, naka-twist siya and now in DNA replication, kailangan mo siya i-open up. So, kapag ino-open mo yung DNA mo, magkakaroon ka ng problem. Kasi, habang ino-open mo yung, wait lang, may nag-raise ng hand. Yes po. May tanong po ba? Ah, ah, may tanong ba? Or something? Wala naman. Nasaan na ako? Wait, sorry. So, di ba, again, your DNA is a twisted ladder. So, kapag ino-open mo yung DNA mo, pag i-replicate mo yung DNA mo, kailangan mo siya i-open. So, imaginein mo, nag-o-open ka ng rope na ganito, ng lubid na ganyan, na naka-twisted, tapos kailangan mo i-open. And it will come a time na yung kapag habang ino-open mo yung lubid, yung lubid mo, hindi mo na siya ma-open. Why? Magkakaroon ka ng torsional stress dito. 'Di ba? Hihigpit ng hihigpit ng hihigpit. Hihigpit ng hihigpit. So, ang solution ay yung DNA topoisomerase. And DNA topoisomerase, it solves the winding problem that arises during DNA replication. So again, ito 'di ba yung DNA mo, and when you open your DNA, there will come a time that it cannot rotate anymore because of the torsional stress, right? And sometimes magkakaroon ka pa ng supercoiling. So paano mo na ma-replicate yung DNA mo kung may supercoiling tas di mo na ma-open? So yung DNA topoisomerase mo yung nagso-solve ng problem na yon. And manonood tayo ng video. Yay!
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Naka-mute pala ako. So, 'di ba, nakita niyo ba yung video ng Clear? So, doon sa video, 'di ba, pinakita na, when you unwind your DNA, magkakaroon ka ng torsional stress. And so, topoisomerase, there are two types, the topoisomerase 1 and the topoisomerase 2. So, yung topoisomerase 1 mo raw, kina-cut niya lang yung isang strand, then niro-rotate niya para mag-open. Okay? Yung topoisomerase 2 naman, kina-cut niya both strands. And then, parang pinapalabas niya yung isang strand doon na naipit sa gitna and maaalis niya yung supercoiling. Okay? So, saan na? So, ngayon, um, your topoisomerase now can solve the winding problem that arises during DNA replication. So, ayun. And yung counterpart again na character nito sa prokaryotes ay yung DNA gyrase. And essentially, same lang yung function nila. So, isipin nyo lang yung function ng topoisomerase and ng gyrase in bacteria is to solve the winding problem during DNA replication. Parang inaalis nila yung stress doon sa strand. Okay? Now, the next character that is involved in unwinding in the DNA replication fork is the DNA helicase. And ito yung pinaka-popular. Pinaka-popular kasi maraming ginagamit to sa pickup lines. Pero di ko na sasabihin sa inyo. I-find out nyo na lang. So, kapag basta. So, DNA helicase, it unzips the DNA double helix. So, for you to remember DNA helicase, isipin nyo lang yung zipper. Okay? So, yung DNA helicase mo, it separates the two DNA strands. And ito yung itsura niya actually. Um, ito yung itsura niya. And what it does is that it binds to your DNA strand and it opens up your DNA. So, ganito yung itsura niya para i-open up niya yung DNA mo. So, yun lang yung DNA helicase. So, remember nyo lang na yung DNA helicase, ang function niya ay to unzip your DNA. Okay? And the next protein involved is your single-stranded DNA binding proteins or SSBs. So, bakit ba important ang SSB? So, let's take a look at this figure. So, when your DNA helicase opens up your DNA, merong tendency na yung DNA strands mo, they anneal to each other or magdikit-dikit sila. Okay? So, for us to prevent that, kailangan mo ngayon ng single-strand binding proteins, itong heart shape dito. And yung ginagawa nila, tinutulungan lang naman nila na hawakan yung strand mo. So, parang yung role lang nila doon sa drama mo, supporting character lang. Parang tagahawak lang nung DNA strands mo. Okay? Para hindi sila magdikit-dikit. Kasi again, if magdikit-dikit yung DNA mo dito, hindi mo mare-replicate ng maayos, right? So, kailangan mo na naka-stretch yung DNA mo. So, therefore, you need your single-strand binding protein. Okay? So, ayun. So, review lang natin yung tatlo na nakita na natin. First is yung topoisomerase, which unwinds your DNA. And gyrase, yung counterpart niya sa bacteria. And next is yung helicase, which unzips your DNA. It separates the two DNA strands. And lastly is your single-stranded DNA binding proteins, or SSBs. They prevent your DNA strands from re-annealing. They support your DNA strands, okay? Para hindi ito magdikit ulit. Okay. So, the next step for DNA replication is for you to add a primer. So, what is a primer? It is a sequence of RNA synthesized by primase and it is needed before the DNA polymerase can add nucleotides to the daughter strand. So, binasa ko lang, di ba? So, primer, isipin nyo, sa mga nagme-makeup, di ba? Bago ka mag-makeup, kailangan mo ng primer. So, kailangan mo muna start, nung start na yung primer mo basically, nag siya yung nagsasabi kung saan magse-start ka or saan ka magsisimula ng DNA replication. Okay? So, ito yung itsura niya. So, ito yung primase mo. Primase raw yung enzyme na gumagawa ng primer. So, your primers are short strand of RNA sequences, usually 10 nucleotides long. So, maikli lang yan, 10 nucleotides long lang yan. And gawa siya sa RNA. And this RNA primer, it signals or it points out where your DNA synthesis will start. Okay, so ayun. So, if you take a closer look, ito yung RNA primer mo, right? And now, since meron ka ng primer, pwede ka na mag-add sa 3' end ng DNA mo, ng nucleotides mo. Okay? And malalaman nyo tong, huwag kayong ma-intimidate dito sa structure na to. I-explain ko yan mamaya. Ito. Yan. Okay, okay. So, ito, di ba, gets nyo na yung structure ng DNA. So, para di kayo malito kapag may mga gantong structures, okay? And ito yung deoxynucleoside triphosphate, i-discuss ko mamaya. Yan yung primer. Wait, tingnan natin. Yung primase yung enzyme, right? And what it does is it adds RNA bases, which are usually 10 nucleotides long, and that is your primer. Okay. So, RNA primer and not DNA primer because, 'di ba? Bakit DNA replication yung ginagawa natin? Bakit ka maglalagay ng RNA bigla? Parang, ha? Para namang ewan kasi bakit gano'n, 'di ba? Nage-gets niyo ba yung feeling ko? Parang nafu-frustrate ako. Bakit kaya dinesign ng Diyos na RNA primer yung ilagay, hindi na lang DNA primer agad-agad, 'di ba? Kasi DNA naman yung goal natin. Bakit RNA primer pa? So, bakit nga ba? If you use RNA as a primer, it automatically makes the RNA sequence suspicious, thus making it easier to proofread. Kasi, yung in DNA replication, merong ability yung isang enzyme, which is DNA polymerase, to proofread itself. Okay. So, suspicious na kapag RNA primer mo, RNA yung gamitin mong primer. So, parang impostor siya dun sa sequence mo. So, kapag may impostor dun sa sequence mo, mas madali na mag-proofread na parang, wow, impostor yan, alisin natin. So, mapuproofread niya na ng maayos yung DNA strand mo. Okay? That is why we use a RNA primer instead of a DNA primer kasi it automatically makes your RNA sequences suspicious, thus making it easier to proofread. Kasi later on, makikita nyo, merong ability yung DNA polymerase natin to proofread itself. Okay? So, let's take a look at elongation. So, ito na, DNA polymerase. Okay, ito yung parang bida sa drama natin kasi siya yung responsible for adding new nucleotides in the growing DNA strand. Ito yung main character natin, yung pinakahintay natin, si Gumjandi, si Sunshine. Okay, siya yung Sunshine. So, ito yung substrates niya are called deoxyribonucleoside triphosphates, or for short, DNTPs. So, remember in your chemistry class, when you have an enzyme, you have a substrate, right? Chemistry class. So, yung enzyme mo dito, DNA polymerase. And yung substrate mo ay yung DNTPs, or deoxyribonucleoside triphosphates. Ito yung nakita nyo kanina. So, therefore, DNA polymerase, it acts on the DNTPs. And what it does is that, ito yung DNA polymerase mo, yung green, it adds the DNTPs in your growing DNA strand. Okay? So, DNTP or deoxyribonucleoside triphosphate kasi, tingnan nyo, tatlong phosphate groups, triphosphate. Deoxyribonucleoside dahil deoxyribose yung sugar and meron kang nucleotide base, itong maliit na blue. Okay? So, yun lang yung meaning behind the name. So, now your DNA polymerase, it adds your deoxyribonucleoside triphosphates in the growing DNA strand. So, nag-grow yan. Okay? Yan yung ginagawa ng DNA polymerase. And actually, for you, di ba, remember nyo the DNA structure? Yung nucleotide, one phosphate group, one sugar, one nucleotide base, right? So, yung sa deoxyribonucleoside triphosphates mo, dalawa yung phosphate group. So, ngayon, yung DNA polymerase mo, tinatanggal niya yung dalawang phosphate group para isa lang yung phosphate group na ma-add. Okay? Makikita natin yan mamaya. So, ito, tingnan niyo. Para di lang kayo puro imagine, di ba? So, yan, merong mga animations na available online. Yung DNA polymerase, ito, ina-add niya yung bases. Okay. Ano bang nangyari? Ayan. And in the leading strand, the addition of the bases is continuous. Okay? Remember, kasi, again, um, anti-parallel yung DNA strands mo and meron kang leading and lagging strand. And in the leading strand, continuous yung synthesis ng DNA mo. Okay? So ito, in-depth, tingnan natin in a closer way yung pag-add ng deoxyribonucleotide. So, di ba? Ito ay yung DNTP, deoxyribonucleoside triphosphate. Why? Again, triphosphate. Tatlong phosphate groups, isang sugar, isang base. Okay? So, yan yung substrate na ginagamit again ng DNA polymerase mo. So, how is it added? Diba? Your DNA grows from 5' to 3'. And in your 3' end, meron kang hydroxyl group. And this hydroxyl group is very significant. Why? Because it acts, nagkakaroon siya ng nucleophilic attack here in your DNTP. So if you remember in your chemistry class, right, nucleophilic attack, basically, ina-attack niya itong phosphate group dito. So therefore, once this hydroxyl group attacks this phosphate group dito, makakat yung phosphate group, magkakaroon ka na lang ng one phosphate group, and yung two phosphate groups mo ma-release in the form of pyrophosphate. And um, this is a favorable chemical reaction. How will you know? Diba sa chemistry class nyo, remember, a favorable chemical reaction, nag-release ng energy. Merong release. Merong release ng energy. And yung energy dito in the form of pyrophosphate. Okay? So, favorable parate na mag-add ka ng new deoxyribonucleotide in the 3' end. Why? Because there is a presence of the hydroxyl group which act nucleophilic attack in your DNTP. And if you observe your 5' end dito, wala siyang OH group. So therefore, wala kang pang nucleophilic attack. So kaya dahil sa 3' end lang nag-a-add ang deoxyribonucleotide dahil sa 3' end again, meron kang hydroxyl group na nagno-nucleophilic attack sa iyong DNTP. And in the 5' end, wala kang hydroxyl group. And this reaction is favorable because, again, it releases energy in the form of pyrophosphate. Okay? Okay. Pag may questions, mag-raise lang kayo ng hand. Okay? Next. Next na support character natin is called the sliding clamp and the clamp loader. So, support to dahil accessory protein lang siya which helps the DNA polymerase to hold on to the DNA strand. So, bakit ba? Kailangan 'to kasi kapag pinabayaan mo yung DNA polymerase mo mag-isa, mahuhulog siya. Yun yung reason behind. So, ayaw mo naman na mahulog agad-agad yung DNA polymerase mo, right? Kasi yung DNA polymerase mo, siya yung bida. Siya yung responsible for synthesizing a new DNA strand, right? So, now your sliding clamp, it acts as support. So, let's take a look at this figure. So, meron daw clamp loader. Parang ito yung support ng support, ganun. Basta. Ito yung support ng supporting character which is your sliding clamp. So, your clamp loader mo, ilo-load niya yung um, sliding clamp in the DNA strand. So, take a look here. So, dito sa part na to. Ito yung DNA strand mo, right? And now, you have your sliding clamp here. Now, once your sliding clamp is here, your DNA polymerase can bind to the sliding clamp. And this prevents the DNA polymerase from falling out of the DNA strand. Siya yung supporting character ng DNA polymerase. Okay, yun lang yun. So now, 'di ba sinabi ko, merong proofreading mechanism yung DNA polymerase. So ito, nakita niyo na 'tong figure na 'to, right? And again, DNA polymerase, what it does is that it adds new nucleotides to the growing DNA strand, right? But it also has a proofreading mechanism. So how? If the correct deoxyribonucleoside triphosphate is added, it will tighten its grip. So for example, yung base mo dito ay adenine and yung parating na DNTP ay thymine. So tama 'di ba? Adenine magbabind sa thymine. So if tama 'yon, yung grip ng DNA polymerase mo magta-tighten. Therefore, adding now your deoxyribonucleotide. So ito 'di ba? Nag-tighten yung grip. But if it is a wrong base, hindi siya magta-tighten. So DNA polymerase tightens its grip for the right nucleotide to be added. Okay? Yan yung one proofreading mechanism ng DNA polymerase. Meron pang isa. And that is exonucleolytic proofreading by DNA polymerase. So, actually, there are two regions in your DNA polymerase. First is your polymerizing region. So this region is where your DNTPs are added, your nucleotides are added. And your E region is where it is edited. So if ever there is a wrong nucleotide base added, your DNA polymerase has a region where it edits your nucleotide. Okay? And now, this causes the mutation rate for us to be low. So, bakit ba natin inaral yung mutation rate nung una, 'di ba? Kasi pinapakita dito na merong mechanism yung DNA polymerase na mag-cause na mutation rate natin ay low. So, kaya hindi tayo agad-agad nagkakaroon ng mutation sa DNA natin dahil yung DNA polymerase kaya niyang ma-correct yung sarili niya. So, 'di ba, sana all self-aware. So, meron siyang self-awareness or proofreading capability na ma-correct niya yung pagkakamali niya. Kahit magkamali siya, kaya niyang i-correct yung sarili niya. So, yon yung dahilan kung bakit yung mutation rate sa atin and even in bacteria is considered to be low. Okay? Yun lang yun. So ito, let's take a closer look. So 'di ba, yung template strand mo adenine and yung right na nucleotide base dapat thymine. But may bida-bidang cytosine dito. So bigla yung cytosine nagbida-bida siya, nag-bind siya dito. And mali 'yun. So ngayon, hindi na makakapag further elongate yung DNA strand mo. Now, yung DNA polymerase mo, meron siyang exonuclease activity which basically it removes the wrong DNA base, the wrong nucleotide. It removes the wrong nucleotide. And so, now, since the wrong nucleotide is removed, you can now proceed, your DNA polymerase can now proceed to add the right nucleotide base. And in this case, it is thymine. Okay? So, what about the lagging strand? So, 'di ba, puro leading strand lang yung na-mention ko. Let's take a closer look at the lagging strand. So, again, the lagging strand is synthesized in segments, right? And therefore, since synthesized siya in segments, there are multiple primers needed. So, gagamit ka ng maraming primers kasi segment siya sinisynthesize. So, maraming primers din yung ginagamit mo. So, one strand is synthesized, magkakaroon ka dito ng nick. And this nick will be sealed by DNA ligase. And also, again, each of these segments are called the Okazaki fragment. Okay? The lagging strand is created at intervals. So let's take a look. That is one Okazaki fragment, right? Magkakaroon ka pa ng primer, bagong primer. Tapos, i-elongate, i-elongate primer, tapos i-elongate siya ng DNA polymerase. Mag-a-add. Tapos yun yung nangyayari every time. So, yun, pinakita ko lang yung animation para di kayo mag-imagine parati. Mhm, mhm. Okay. Yun yung laging strand. Wait lang. Mali yung title ko. Oh no. So, ay hindi. Tama yung title ko. Sorry. Dito na ako. So, now, DNA polymerase has an exonuclease activity. So, bakit? Again, balik muna tayo. Balik. So, di ba, laging strand, meron kang maraming RNA primers. RNA to, RNA yan. Marami kang primers. So now, yung DNA polymerase mo, it has an exonuclease activity, which basically means that it has the ability to remove your RNA primers. So in the case of bacteria, yung DNA polymerase 1 yung nagtatanggal nun and nagre-replace ng RNA with DNA. So in your lagging strand, right, again, you have multiple primers because again, your DNA is synthesized in segments. So itong primers na to, tinatanggal to ng DNA polymerase mo via exonuclease activity. And exonuclease activity again is removal of those spaces. So itong exonuclease activity, i-discuss natin to in depth sa lecture 3. Okay? So now, once your primers are removed, magkakaroon ka ng nick dito or magkakaroon ka ng parang, ano bang tawag dun? Separation, parang hindi pa sila connected. So, yung DNA ligase mo, it seals the gap sa lagging strand, yung mga nicks. So, tingnan natin. Exonuclease activity ng DNA polymerase, tinatanggal niya yung RNA primer, right? And magdadagdag siya ngayon ng bagong bases. Now, yung last mong character ay yung DNA ligase mo. And again, it joins the Okazaki fragments together with phosphodiester bonds. So again, your lagging strand, they are formed in segments, right? And these segments, meron pa yung nicks or meron pang mga separated regions. And those separated regions are joined by the DNA ligase through phosphodiester bonds because yung ano dito, yung nawawala dito ay yung bonds between the sugar phosphate backbone. Ayan. So tingnan natin, ito actually yung itsura niya in sa up close. So sa DNA lagging strand mo, separated itong 5' end sa 3' end mo. So what the DNA ligase does is that it uses ATP. ATP is your energy molecule, right? And it needs for a favorable reaction, you need to release energy, right? So that's why your DNA ligase uses ATP. And now, yung ATP mo ngayon, it provides parang parang ia-attack siya. Ay, wait lang. So yung ATP mo, 'di ba dito magkakaroon ka ng phosphate group, mag-a-attach dito. isa lang yung phosphate na matitira dito and yung dalawang phosphate mare-release. And remember, yung 3' end mo may hydroxyl group. And yung hydroxyl group mo magkakaroon ng nucleophilic attack. So, therefore, it attacks this phosphate group here. So, once it attacks this phosphate group here, magkakaroon ka ulit ng release of energy. And then, it results to the or joining together of the fragments. Then, ulitin ko. So, again, a favorable reaction releases energy. So, yung DNA ligase mo ngayon, gagamit siya ng ATP. And ATP is adenosine triphosphate. Ito yan. So, ito yung itsura ng laging strand mo dun sa part na merong separation. And yung ligase mo, what it does is that it attaches um, yung isa lang na phosphate group dito, releasing two phosphate groups. So, may one phosphate group ka dito. So, therefore, this gives your 5' end the characteristic na pwede siyang enucleophilic attack ng 3' end with the OH group or the hydroxyl. Now, once your hydroxyl group attacks this phosphate group here, magkakaroon ka ngayon ng release of energy by the form of AMP or adenosine monophosphate. And dahil favorable tong reaction na to, magjo-join yung nick mo ngayon. Magko-connect sila. Magkakaroon ng phosphodiester bond. Okay? Okay. So, ganyan lang yung simplified version ng DNA like this. So, kino-connect niya lang yung nick doon sa DNA strand mo. Okay, okay, okay. So, ito. Wow, ito na yung overview ng sa eukaryotes. So, yung drama, remember again, pag mag-aaral kayo ng DNA replication, para lang kayong manunood ng drama. Kasi same lang naman yung characters, same lang yung roles, different lang yung names nila. Okay, di ba? Parang si Gong Jandi sa Boys Over Flowers, pero si Sunshine sa Meet Your Garden. So, dito sa eukaryotes, halos same lang siya sa prokaryotes, pero iba nga lang yung names ng mga ginagamit nila polymerase mainly and also the topoisomerase. And meron din sa eukaryotes na different types of polymerase. Meanwhile, meron sa eukaryotes na different types of polymerase. Meanwhile, in prokaryotes, dalawa lang, DNA polymerase 1 and 3. Pero dito sa eukaryotes, let's take a look. Diba? Polymerase delta, polymerase alpha, polymerase epsilon. Similar lang yung function nila actually. Halos same lang yung function nila. So, ganon. Okay? Pero ito, to simplify, ito yung DNA replication in bacteria. And yan, pwede nyo yan basahin pero makinig muna kayo sa akin please. So, yung first na nangyayari ay, again, review natin, meron kang ORC or Origin of Replication Complex. It is the initiator protein which binds to the origin of the replication. And this part of the DNA is rich in adenines and thymines, right? And why adenines and thymines? Because adenines and thymines has two hydrogen bonds as compared to guanine and cytosine na may three hydrogen bonds. Therefore, adenines and thymines are easier to break. Okay. Now, once you meron ka ng origin of replication, you will form your replication fork. And in your replication fork, you will see your DNA gyrase for bacteria or topoisomerase in eukaryotes. And this solves the unwinding problem in the DNA. It removes the torsional stress when you stretch out your DNA. And next is your helicase. You will see your helicase at it is your enzyme that separates your DNA strands together. It unzips your DNA. And another supporting character is your SSB or single-stranded binding proteins, which supports the each of your DNA strands to prevent them from attaching again from each other or annealing from each other. Okay. Next, to initiate your DNA replication, you have your RNA, you have your primase. And your primase, it synthesizes an RNA primer. And RNA primer is usually 10 nucleotides long. And this marks where your DNA replication starts. Okay. And once you have your RNA primer, your DNA polymerase can now add new nucleotide bases in your growing DNA strand. And in the case of bacteria, you use DNA polymerase 3. And this is your leading strand because the DNA synthesis here is continuous and it is towards the replication fork. And your lagging strand now has multiple primers. It uses multiple primers. And it is made in segments. And ito lang yung DNA polymerase niya. Okay? And ito lang rin yung primase. And the primase, it makes up your primer. Okay? And now that you have multiple primers in your lagging strand, your DNA polymerase can form segments of the DNA, and these are called your Okazaki fragments. Okay. And lastly, once your RNA primers are removed by exonucleolytic activity by the DNA polymerase, um, yung DNA polymerase mo mag-a-add ng new bases in the primer region and the next here in your DNA fragments or DNA segments are connected together by the enzyme called DNA ligase. And yun na yun. So, para ma ah, 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 para mas maintindihan nyo at animation siya, ito. So, panoorin natin yung animation. So, yung animation na nahanap ko na pinaka-complete kasi yung iba minsan kulang-kulang. So, okay.
DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having three phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes two replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing two replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase III is replaced by DNA polymerase I, which has 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
Okay, okay. So, yun yung complete animation ng DNA replication. Yun yung pinaka-complete na nahanap ko. So, meron pa ba kayong question so far bago tayo mag-proceed? May questions pa kayo so far?
Ma'am,
Yes po.
sa ano po, sa polymerase chain reaction, ang ginagamit po, DNA polymerase for
Sa,
yung
pardon?
sa polymerase chain reaction po, ma'am. Yun na po bang DNA polymerase ang gamit?
Good question. Actually, yung polymerase chain reaction, yung PCR, i-discuss natin sa last part ng lecture natin. So, magandang question yan kasi actually yung DNA, yung PCR, yung polymerase chain reaction, it uses the principles behind DNA replication. And tama yung sinabi ni Leila na yung ginagamit actually ay yung DNA polymerase. Actually, yung ginagamit ay DNA polymerase, primers, and DNTPs. Okay, yung tatlong yon yung commonly ginagamit sa PCR. And i-discuss natin mamaya. Meron pa ba kayong question so far? Raise your hand po if meron. Eh, so far, so
Last na,
far.
ma'am.
Sige, sige, go lang.
May ano po ba? May rate po ang DNA replication, halimbawa 50 bases or 100 bases per second na nagagawa.
Okay. So, actually, mabilis ang DNA replication and minsan, ang rate niya sa lagging strand ay 1,000 nucleotide base ng per second or mas mabilis pa ata do'n. Basta gano'n, di rin ako sure sa figures pero search nyo na lang. Pero sa tingin ko sobrang bilis niya. So like, 'di ba yung ano niyo, yung cell division niyo, it can occur within 24 hours. So, imaginein mo yung DNA replication din, sa fraction ng time na yon sobrang bilis lang. Kasi hindi naman sa buong cell division mo magdi-DNA replication ka, sa S-phase ka lang magdi-DNA replication. And mabilis lang siya actually. Pero di ako sure sa figures, mga 1,000 nucleotide bases. Pero i-confirm niyo na lang.
Okay
Okay?
po, ma'am. Thank you po.
Okay, okay. Meron pa
Ma'am,
bang question? Ah, yes po. Yes po.
pwede ma'am yung Okazaki fragments medyo elaborate patungkol po dun sa yung sa nick po sa laging strand ganun. Di ko masyadong
Ah, okay.
nakuha.
Balik muna tayo sa Okazaki fragment. So, mhm. Ito, um, yung Okazaki fragment mo, yun yung tawag sa fragment, sa segments in your lagging strand. Yun lang yung tawag basically sa segments of DNA synthesized in your lagging strand. Yun yung Okazaki fragments. And named as such because small history lang, yung nakadiscover ng Okazaki fragments ay couple named na ang ay Okazaki. So, 'di ba, in your leading strand, madali lang intindihin kasi continuous lang yung DNA replication. And in your lagging strand, different kasi nga anti-parallel yung DNA strands mo, right? So, therefore, different din yung direction nung pag-add mo ng new nucleotide bases in your new DNA strand. Okay? So, ngayon, when you make your Okazaki fragment, dito sa part na 'to, 'di ba gumagamit ka ng multiple primers, right? To form different Okazaki fragments. And now, you need to remove these primers kasi again, ang ginagawa mo DNA, hindi RNA. So you have to remove these RNA primers. And when you remove these RNA primers, nag-a-add dito ngayon sa ah, kapag tinanggal mo na 'to, mag-a-add again yung DNA polymerase mo ng new nucleotide bases. And while it adds nucleotide bases, nagkakaroon ka ng nicks doon sa portion ng end ng strand ng each Okazaki fragment mo. So, these ends of the Okazaki fragments are joined together by your DNA ligase. And again, yung way on how the DNA ligase joins these strands together is via phosphodiester bonds. And again, your DNA ligase uses ATP. And two of those phosphate groups are released. So, meron ka na lang ditong one phosphate group or this is called adenosine monophosphate. And now, since may hydroxyl group yung 3' end mo dito, meron siyang ability to uh, nucleophilic attack itong phosphate group mo dito. And that is favorable again because there is a release of energy and every favorable reaction, meron kang release of energy. And once that happens, yung um, yung section mo now dito can form phosphodiester bond. Nawala kayo. Clear ba yun?
Okay na po. Thank you po.
Okay, okay, okay. Okay. Wait, 11:25 na pala. Oh no. Okay. So, proceed na muna tayo sa termination. So, last, diba, termination. So, nakita nyo sa video na yung sa bacteria, madali lang ang termination. Kasi circular lang yung DNA sa bacteria. And by the end of the replication in bacteria, basically, isi-separate niya lang yung two circular DNA. And yung two circular DNA mo na yon mabibigay niya sa two daughter cells niya through binary fission, 'di ba? Yung cell division sa bacteria is called binary fission. So, yun. Ayun lang, simple lang siya sa circular DNA. But in eukaryotes, meron tayong problema. So remember, eukaryotes is linear DNA Yung sa atin, linear, so you have to stretch it. And that is called the end replication problem. And the end replication problem, for a linear DNA molecule such as that of the eukaryotic chromosomes, the usual DNA replication machinery is unable to replicate the end. So, tingnan natin. 'Di ba? Um, Meron tayong lagging strand. Wait. So first, magkakaroon tayo ng DNA replication dito. So, 'di ba meron tayong sinasabing lagging strand? And yung lagging strand mo, kailangan mo ng multiple primers. Okay? But finally, one primer remains on each of the daughter DNA molecule in the lagging strand. So, sa end nito, meron kang matitirang primer. And of course, these primers are removed. So, therefore, magkakaroon ka ng gaps. And problem yon because this now, since magkakaroon ka ng gaps, therefore, mas iikli yung DNA mo. So, now, every time your DNA replicates, umiikli siya ng umiikli actually. So, as you age, mas umiikli yung DNA mo. Okay? So, yun yung problem sa eukaryotes. Kasi, habang nagre-replicate yung DNA mo, umiikli siya ng umiikli. Okay? Now, to solve that, we have telomeres. And na-amaze ako sa telomeres kasi na-discover lang siya recently, mga 2009, ganun. Or yung telomerase enzyme pala, basta recently lang. And telomeres are highly repeating DNA sequences at the terminal end of the chromosomes. So, telomeres are highly repeating sequences. And they form as caps at the ends of the chromosomes. And unique sila kasi repeating sequences sila and non-coding sila. So, hindi sila nagko-code for protein. But, uh, they provide a protective mechanism for your chromosomes because they are repeating sequences. So, as you can see here, yung repeating sequence dito sa tetrahymena ay GGGTT, GGGTT. Repeating sequences lang yan, yung telomeres. And your telomeres, they shorten until cell division stops. So, yung parent cell mo, normal pa yung telomeres niya. Yung daughter cells niya, mas umikli. Yung daughter cells nito, mas umikli. Yung daughter cells nito, mas umikli yung telomeres mo. Hanggang sa mag-stop na siya. And in this phase, delikado. Kasi since wala ka ng telomeres, yung makakat mo na DNA ay coding DNA, right? And they are essential. So, kapag nakat to dito, pwede kang magkaroon ng mga sakit. Okay? So, how do we solve this? So, telomere replication by telomerase. So, yung ginagawa lang ay ine-elongate yung repeating sequences. So, again, this is your telomere repeating sequences by the end of your chromosomes or your DNA. And now, this is your telomerase. It is an enzyme which adds new nucleotides here by the end of your telomere. So, ine-elongate niya to. So, look here, na-elongate niya. Therefore, when this is elongated, yung DNA polymerase mo now can add new strand here. So, makakapag-add ka pa rin dito kasi mas elongated na to. So, that is how the shortening of the DNA is um, solved. Okay. So, ito lang yung telomerase mo. Again, ito yung telomeres mo, repeating sequences, and ine-elongate niya lang sa end ng DNA mo. And also, mayroong T-loop na nakikita sa end ng mammalian chromosomes. And this T-loop, um, inevitably kasi 'pag nagre-replicate ka ng DNA, magkakaroon ka talaga ng overhang or ng space. So now, this overhang, it tucks itself between two strands, the two strands of your DNA in your telomere region to protect it. Because if you don't protect this, some enzymes might act on this on this overhang tapos iisipin nila foreign body yon or ia-attack nila and pwede kang magkaroon ng sakit. And yon. So what your chromosomes does is that it tucks this overhang in between your DNA strands here in your telomere region. So therefore, magkakaroon ka ng T-loop. Okay? And for our case study, we look at telomerase and cancer. So, actually, sobrang interesting nito para sa akin. Kasi sa cancer, meron kang high, anong term? Meron kang parang high expression ng telomerase. And telomerase again, it extends your telomere region, right? So in cancer daw, yung telomerase mo, yung enzyme mo, merong high expression, hyperactive. So let's take a look. So again, as your cells divide, your telomeres shorten. And there are checkpoints in your cell division, which we will discuss in depth in our future lecture. So, um, if your cells they pass through these checkpoints, nakapass through sila na hindi sila na-check, pwede ka magkaroon ng cancer. So, unchecked cells ito. And now these cells, they continue to divide by using telomerase. And telomerase, they lengthen your telomere sequences. And your cancer cells, hyperactive yung telomerase mo. So, magde-divide lang ng magde-divide yung DNA mo na hindi nagso-shorten yung telomere ends mo, na hindi nagso-shorten yung DNA mo. So, infinite lang siya ng nagde-divide kasi again, yung telomerase mo, siya yung nagpe-prevent na mag-shorten yung DNA mo. Nagigets niyo ba? So, yun. So, parang yung telomerase din, ano siya, Um, sagot din siya for anti-aging actually. Sagot din siya for anti-aging. Pero kapag nasobrahan, kapag hindi mo siya na-control, pwede ka magkaroon ng cancer because again, your cancer cells, they take advantage of the fact that your telomerase, they maintain your telomere sequences and therefore your cells can divide without your DNA sequences shortening. Okay? So yun. So yun lang yung sa DNA replication. And um, ito na, we studied the mechanism behind DNA replication because ito yung tinanong kanina ni Leila. Not only to know how cells pass on their hereditary information, but also to learn the principles behind techniques in molecular biology. And nakaka-relate ako kay Kim Chu habang nagpabasa nito. So, ito, PCR chain reaction. So, ito na yung application ng natutunan natin. Yay! So, PCR or polymerase chain reaction, di ba pinakita ko na sa inyo yung sa COVID lab, yung ginagamit para ma-detect yung RNA ng yung virus, yung coronavirus, gumagamit ng PCR. So, yung behind dito ay actually, merong gantong stages. Pero dito muna tayo. So dito, sa tube na 'to, yung PCR ganito yung itsura, yung PCR machine. Dito mo ilalagay yung samples mo. Para lang siyang, magbe-bake ka pero sobrang liit. Parang lang siyang oven pero sobrang liit. Tas microtubes yung gagamitin mo. And dito mo titingnan or i-a-adjust yung conditions, which are temperatures. Temperatures yung ina-adjust mo dito. So in this tube, you put here your sample, uh, your DNA polymerase, your DNTPs, and your primers. Okay? So why? Yung sample mo, it acts as a template. A template, yun yung template na gagayahin mo. And yung DNA polymerase mo, and yung primers mo muna, yung primers mo, they initiate yung replication, right? Now, yung DNA polymerase mo now adds yung nucleotide bases. Okay? Nalimutan ba ako? Yun. So, ito yung steps behind polymerase chain reaction. So, meron kang double-stranded DNA. And there are 3 basic steps. Madali lang to, promise. Ito yung denaturating stage, annealing, and extension. So, in denaturation stage, yung double-stranded DNA mo, isi-separate mo sila. So, kaya mo i-adjust yung temperature sa PCR machine mo kasi at a certain temperatures, yun yung stages mo. So, for the denaturation stage, usually 94 to 95 degrees Celsius. Yun yung time kung saan magse-separate yung DNA mo. So, DNA strands mo. And next is yung annealing stage. So, annealing meaning connecting or didikit siya. So, you have your primers and your primers, they anneal to your template strand. And then, they mark where the DNA replicate or the replication should start. And now, in this annealing stage, it usually happens at 50 to 56 degrees Celsius. And your last stage now is your extending stage or your extension stage where your DNA polymerase acts. So, yung DNA polymerase mo dito, magdadagdag na siya ng nucleotide. Okay? So, baka tanungin niyo, bakit iba-iba yung temperature? 'Di ba? Bakit kaya iba-iba yung temperature for each stage? Dahil ang enzymes natin, some enzymes, they act at optimal in certain temperatures. So, for example, yung primase mo, yung primers mo, optimal sila at this temperature. And yung DNA polymerase mo, optimal siyang nag-a-act at this temperature, at 72 degrees Celsius. Okay? But actually, these temperatures are only examples because when you do an actual PCR, you have to optimize yung experiment mo. I-optimize mo yung temperatures mo. So, paulit-ulit ka ng paulit-ulit hanggang sa mahanap mo yung optimal temperatures mo. Depende rin yon sa samples mo, depende rin yon sa primers mo, and depende rin yon sa DNA polymerase na gagamitin mo. And most of the PCR reagents that you use, mabibili mo na siya. Meron kang kit. And yung kailangan mo lang actually dito na i-design ay yung primers mo. Kasi different din yung parang different din yung gusto natin i-detect, 'di ba? Na DNA segment. So, kailangan mo i-design yung primer mo na sasakto dun sa segment na gusto mo i-amplify. So, yung PCR, i-discuss natin 'to ng mas maganda in your laboratory. Pero ito lang yung overview na. Okay. So, ito yung nangyayari sa polymerase chain reaction. So, you have your template DNA here, and for each cycle, paulit-ulit lang yan na temperature changes, na-amplify mo ngayon yung DNA mo. So, amplification. So, dyan, ang principle behind dyan, again, ay DNA replication. Okay?
Ma'am,
Yes po.
nag-post ka ma'am ng quiz po. May time limit po yun?
Yes. Wala yung time limit. Okay. Oh no, nakita niyo na yung quiz ko. Pero, tapusin muna natin to. Pero, tinatawa na naman ng kapatid ko. Tapusin muna natin to. Bibigyan ko kayo ng instruction sa quiz. Huwag kayo mabahala. Okay? Relax lang.
Ma'am?
Okay. Yes po. Yes po.
Ma'am, wala pa po ako sa Google Classroom po.
Inad na kita. Late ko nabasa yung message mo. Sorry. Pero inad na kita sa Google Classroom. I-check mo ulit. Okay?
Okay po, ma'am.
Okay. So, to summarize, we learned about the mechanism behind DNA replication. And we identified the different enzymes and proteins behind DNA replication. And we also learned that humans and bacteria have relatively low mutation rates because of the proofreading mechanism by the DNA polymerase. And also, we learned about the principle behind PCR and how it is used, or what and it uses the mechanism of DNA replication. Okay. So, now that we've learned about DNA replication, ito na yung central dogma of molecular biology again. Naaral na natin yung DNA structure, naaral natin yung hereditary information, naaral na natin yung DNA replication in depth. So, DNA replication again is how our cells replicate our own DNA. Yun na yun. So, it yun lang, and may questions ba kayo bago tayo mag-quiz? May questions ba kayo? So kung wala pa kayo sa Google Class, i-send ko na lang yung link ng quiz sa GC, sa Facebook group. And yung quiz, actually, tungkol siya sa lecture 1. Wala naman yung time limit. Actually, magiging maluwag na ako sa mga requirements. So, lahat ng requirements na ibibigay ko, lahat yon, yung deadline ay last week ng SEM na to. Dahil alam nyo naman, iba yung panahon natin, pandemic. Yeah. And pero mas maganda kung on time nyo pa rin ipasa para meron kayong sense na meron kayong progress, di ba? So maganda pa rin kung on time. Pero since nga iba yung panahon natin, last week ng SEM na to yung deadline ng lahat ng mga requirements natin. Okay? May tanong ba kayo sa DNA replication?
Ma'am?
Ma'am, may tanong
Yes.
po.
Ay, sige, ikaw muna,
Ay.
Jeff.
Ma'am, since nabanggit niyo po, ma'am, yung sa cancer po, ma'am, dun sa extension po, ma'am, ng abnormal replication po, ma'am, sa telomeres. May tanong lang po ako about karyotyping. Magre-reflect po ba, ma'am, yung dun? Yung magiging abnormal po yung chromosome?
In karyotyping, yung telomeres, yung tanong mo ba kung makikita mo yung telomeres sa karyotyping? Or kung makikita mo kung nag-shorten?
Opo.
Okay. Actually, pwede mo siyang makita. Actually, interesting tong research. Bibigyan kita ng idea para ma-research mo balang araw. Like, pwede mong tawag dyan, pwede mo i-compare yung telomere length ng tao na may cancer and tao na normal by pwede mong lagyan actually ng fluorescent probes yung telomeres mo. So fluorescent probes umiilaw siya. So kapag nilagyan mo ng fluorescent probes yung telomeres mo habang nagka-karyotyping ka, pwede mong makita yung difference ng telomere length between tao na normal and tao na may cancer. And makikita mo, mas mahaba yung telomere length sa may cancer. Kasi again, overexpressed yung telomerase sa may mga cancer. And for normal people, sa atin, yung telomere length natin umiikli ng umiikli habang nag-age tayo. So, maganda siya actually na research topic. And yung tanong mo again ay kung makikita mo yung telomeres mo yeah, habang nagka-karyotyping, yes, you can by using fluorescent probes. Pwede mo siyang kulayan or pwede mo siyang ilawan. Meron pa bang tanong?
Ma'am, sa retrovirus po, sa DNA po ba yan nag-aatake sila? Since RNA po baga, ma'am. Tapos may, ano po, sa, naapektan po yan ng, replication?
Oh, in retroviruses, wow, advanced. So, ano natin to, mga third part pa ng lecture. Pero yung viruses, they use the replication machinery of our cells. They hijack our cells and they trick us to use the replication machinery. So, i-infect nila yung cells natin and gagamitin nila yung replication machinery ng cells natin. And minsan, they actually parang ini-incorporate nila yung 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 hereditary information nila minsan sa sarili nating hereditary information. So makikita mo yon actually mostly in bacteria. So when a virus infects a bacteria, minsan in-incorporate yung virus yung hereditary information niya sa bacteria and then yung bacteria mo now it will use its own replication machinery to then multiply the viruses. Pero yung viruses, mas i-explain pa natin yan sa ating third part sa may part ng infectious molecular biology of infection. Sa may third part, second to the last ata na lecture. Ganun. Or third to the last. Pero good question.
Thank you po, ma'am.
Okay. May tanong pa ba? Bago tayo mag-end. At nataranta na sila sa quiz. Okay. So, kung wala na kayong question, nag-post ako ng quiz sa lecture 1. Madali lang yon, sobrang dali lang, promise. And meron ulit na class participation. Kung may naisip kayong tanong, tanungin nyo doon. Dahil yung lahat ng questions nyo, kino-compile ko yon and sasagutan natin bago mag-periodic test. Lahat ng questions nyo. So, kung
Ma'am,
wala
may
ng
tanong.
tanong, mayroon pang
Ay,
tanong. Meron pang tanong.
sige. Ano, ma'am, gusto ko lang malaman, ma'am, sa ano, yung confirmatory testing, ma'am, sa HIV, yung PCR po ng HIV. Paano po yung mechanism niya? Like yung sa viruses,
Oh,
ganun. Same lang po ba yung sa retrovirus sinabi na Layla?
halos same lang. Yung sa PCR pa. Okay.
Di ba
So,
PCR
balik
po
tayo.
sa HIV?
Actually, yung polymerase chain reaction, yung mechanism niya for both DNA and RNA, same lang. Yung pagkakaiba lang ay yung sa denaturation stage kasi di mo naman kailangan i-denaturate yung RNA mo kasi single strand lang sila, di ba? So, sa pagkakaiba lang ng DNA and pagkakaiba lang ng kapag i-detect mo yung virus, for example, yung coronavirus, um, yun sa denaturation stage. And magpo-proceed ka dito sa annealing stage kung saan i-design mo yung primers. And paano ba? Yung sa example ng PCR for DNA, for double-stranded DNA, yung ginagamit ay yung BCR-ABL. Alam niyo ba yung BCR-ABL? Sakit siya na leukemia. So, yung dalawang sections mo ng chromosome, nagkapalit siya nag-exchange sila ng position. So, yung BCR gene mo, nag-exchange ng position sa ABL gene mo. Pero, i-search nyo na lang. Pero, yun yung parang example kung saan ginagamit yung PCR sa double-stranded DNA. Yung BCR-ABL, yung case, leukemia siya, sakit siya, which is leukemia. And sa virus naman, such as coronavirus, since RNA, single strand lang, yung ginagamit ay yung magde-design ka lang ng primers which binds to a specific part of your RNA and um, yun na yung parang start kung para mag-extend yung dito. And pero actually different types ng PCR. Yung na-mention ko 'di ba yung RT-PCR, yung real-time PCR. Meron rin yung sinasabi na um yung yung conventional which is gagamit ka pa ng agarose gel electrophoresis, yun. And meron rin na gumagamit nung anong tawag dun? Parang from RNA gagawin mong DNA tapos DNA saka mo siya i-amplify. Ano ang tawag dun? Basta yon. Sasabihin ko na lang sa inyo pag nahanap ko ulit siya. Nawala na yung word sa isip ko. And ano pa bang share ko tungkol sa PCR? Yun lang. So, meron pa ba kayong tanong? Ano nga yun? Meron pa ba kayong tanong? Or something? Okay, okay. So, reverse transcriptase PCR. So, yun yung nire-reverse transcription niya. So, tingnan natin dito sa Central Dogma of Molecular Biology. Oh, wait lang. So, yung sa reverse transcriptase PCR, basically, gagawa ka ng DNA from RNA. So, yun, isa rin yung type ng PCR. So, from RNA to DNA. So, parang yun ba yung sinasabi ni Leila? Na ngayon ko lang na-get na para yung virus mo pwede mo siyang i-amplify yung RNA by reverse transcription. Reverse transcription meaning you reverse this from RNA to DNA. So isa lang yung type ng PCR. So yung other types ng PCR ay yung conventional in which you use agarose gel electrophoresis after you PCR so you can detect the genes amplified and also the real-time PCR which you use a computer. So sa computer makikita mo in real time yung kung na-amplify ba yung gene. Okay? Pero in-depth natin i-discuss yung PCR sa laboratory natin. So meron pa ba kayong questions? So, kung wala ng questions, pwede nyo naman isulat doon sa ating Google Form sa class participation na may 2 questions. Ano yung natutunan nyo? Sana meron naman. At yung pangalawang question ay kung ano yung questions nyo. And yung questions nyo, i-compile natin siya. And also, na-orient na ako ni Sir Isan na lahat ng classes natin ngayon ay lecture muna. Tapusin daw muna yung lecture. Tapos yung laboratory classes daw sa end na lang daw ng SEM sa May. So, sa Monday, lecture tayo. And yung topic natin ay DNA repair and recombination. And may quiz ulit para sa lecture 2. Okay, para ma-review kayo before your long test. So, mas maganda, di ba, na small chunks of information na re-review. So, yon. And ano pa ba ang aking announcement? Meron kayong quiz. 8 minutes na lang. Hala. Meron kayong quiz and wala naman siyang deadline. Sagutan nyo yun. Pero mas maganda kung sagutan nyo siya on time para may sense kayo ng progress. And meron pa akong pinost na class participation. So, 2 yung questions again. Sagutan nyo yun. And yun lang. Thank you for listening to me. Goodbye. Mag-lunch na kayo. Okay, goodbye.
Thank you. Bye,
Bye,
ma'am.
ma'am.
Thank you, ma'am.
Thank you,
Okay,
ma'am.
ma'am.
Thank you
Thank
po.
you, ma'am.
Bye, ma'am. Thank you po.
Thank you po, ma'am.
Sure, sure.